Hey guys, this is Mrs. Hallman, and this is AP Bio Cell Cycle Disorders, part of our cell division chapter. This is the first video of a couple that's going to cover this last section of our cell division unit. So let's get started. There are two different types of cell cycle disorders, and they kind of have contrasting issues. The first is when we have cells that divide normally, but they will not stop, and they divide out of control, and that would be cancer. And the second is when we have cells that we would rather have them divide and they cannot divide. So examples of that would be cells that we need to replace or heal, different injuries such as brain or spinal cord injuries. And that leads us to the application of stem cells and how their therapy might be the answer to some of these disorders. In normal functioning cells, we have internal and external signals that will help regulate and act as checkpoints in our cell division cycle. Internal signals are some such as different things that our cell acquires before it is able to go on to the next phase. For example, even though we have multiple chromosomes that are going through mitosis, if there's one chromosome that fails to connect spindle fibers to the kinetic core, the cell will actually delay an anaphase rather than go through the cell division missing a chromosome or having too many chromosomes. So this is an example of an internal signal that would make sure that cell division didn't happen unless the cell was perfectly ready to divide. Other examples of internal signals we've kind of covered already when we talk about kinases and cyclin, and how CDK is part of that checkpoint in mitosis. We also have external signals called growth factors. And these are kind of external signals that make sure that our cell is completely ready before it's ready to go on to the next phase of mitosis. These are usually proteins that are released either by glands or other cells that will trigger cells to divide. One example of that we're going to see in the next slide is called PDGF, or platelet-derived growth factor. Fun fact, there's another one that's called sonic hedgehog that's released during embryonic development, and it helps trigger cell division as we start to develop our neural system. So PDFG is a growth factor that triggers cells to go through mitosis and to properly divide. So one quick way you can test that is if you take fibroblasts, which are tissues found in our body, and if you would dice them into a bunch of different pieces, suspend them and plate them on a medium, those cells would not proliferate and grow without the presence of PDGF, which you can see in this file here. That when PDGF is available, then those cells will go through mitosis. And that platelet-derived growth factor is actually deposited from platelets in our blood. So if you have an injury, like you scrape your skin because it fell off your bike, your platelets will secrete PDGF in order to help promote those cells around the edges of your wound to start dividing so you can eventually heal that wound. Another example of some external signals that come from outside of the cell that is dividing would be density-dependent inhibition. And this is simply when a cell is in contact with cells on all of the other sides of that cell, it realizes it doesn't have any space to continue division, so it will be inhibited. This is important because most of our tissues grow in layers or strata, and once that tissue layer has become completely or has filled completely up the area that it needs to regenerate, we don't want that tissue continuing to divide if there's no space for it. A second external signal would be anchorage dependent, which means that there needs to be some sort of platform or what we call substratum present in order for that cell to rest upon before it's going to start to divide. And you can see in this image both of those kind of exemplified. The anchorage dependent, simply you need some sort of area for those cells to rest upon before they would be triggered to promote to the next cycle of the cell cycle, next stage of the cell cycle. And density-dependent inhibition, if you have a thin layer, a monolayer of cells, and you remove a few of them, they will only divide to replace those cells that were removed until they run out of room. And that is the way our cells are supposed to behave. Of course, when we have transformation and we develop cancerous cells, they start to lack that normal inhibition, and they will start to divide out of control and to pile up in a three-dimensional form, which we now call tumors. Cancer cells, when we do develop cancer, do not respond normally to normal control mechanisms within our body. So cancer cells, whereas normal cells need growth factors in order to divide, cancer cells might not need those growth factors, or they can create their own growth factors that will promote them to grow out of control. They can also convey a growth factor signal without the presence of the growth factor. So that signal transduction pathway will actually transmit the signal even though there was no ligand to actually trigger that pathway. Or they just might have an abnormal cell cycle control system, meaning one of the mechanisms of the checkpoints that are in place goes haywire, it has some sort of mutation that would disable that mechanism. When we first have cancer develop, we say there's a moment of transformation where we go from having normal cells with normal DNA without mutation, and then we go through a mutation event which would cause that cell to transform and become cancerous. The cancerous cells will then divide and form tumors. We say that when that tumor develops, but it's unable to proliferate and move to a secondary site, that is termed a benign tumor. I always tell my middle school and my freshman students that if you have a benign tumor, most likely you will be fine, unless it interferes with some other tissue or organ, because those tumors will not spread to secondary locations. If they do spread and they invade surrounding tissues, we say that they are malignant tumors, and we say that that act of spreading is called is when the tumors metastasize, or a metastasis when they spread to a secondary location. So basically, the way that that happens is a tumor will develop in one type of tissue from one specific cell, and as it grows, you can see in image 2, it'll start to invade neighboring tissues. This is obviously cancer of the breast, and it's invading the mammary glands that then spreads. And in step 3, you can see that it is carried to secondary locations through either blood vessels or lymph vessels or both. And that's how we'd get that metastatic tumor in a secondary location. We typically describe cancer as being in a stage. You can be diagnosed with like stage 2 breast cancer, stage 3 colon cancer. And those stages vary a little bit depending on which cancer you are diagnosed with. But a lot of it has to do with the layers of tissue that the tumor has invaded. So you can see in stage 1 or stage 0 here, it is a very superficial polyp, whereas stage 1, it has started to invade the surrounding tissue. And stage 2, you can start to see that it is now receiving some nutrients from blood supply. Stage 3, it's become more fully vascularized, and it looks like stage 4, one of those tumors has spread and metastasized to a secondary location. So the stage has to do with how developed that tumor is. That brings us to a fascinating and rather tragic story of a woman named Henrietta Lacks. Her cells actually are known as the immortal cells of HeLa cells. We're going to be talking about this a little bit later in the semester, so I won't spoil everything now. But Henrietta Lacks had cervical cancer. A selection of her tumor was taken without her knowing, and cultured in a lab without her knowing. And for some reason or another, HeLa cells possess the ability to divide without ceasing. Normal cells will divide between 20 and 50 times, and by that time they kind of wear out and they can't divide any more than that in culture, which proposes an issue for scientists who really want to do experiments and test with potential vaccines or medicines on real human tissue. If we can't grow tissue in a lab that can last more than 20 to 50 cell divisions, it becomes really difficult for us to gain enough tissue and to have tissue that is genetically the same to act as a constant in an experiment. So enter HeLa. Her cells, for some reason or another, and we have several ideas of why this is, her cancerous cells are able to repair themselves and to divide in culture constantly. In fact, her cells were taken from her in the 1950s, and there are still her cells that are living in literally millions of laboratories throughout the nation and throughout the world. Her cells have contributed towards some of the most major medical advances that we've had in this century. Her cells have gone into numerous cancer treatments. They helped develop the polio vaccine, and just so many other things. And the fun part is we might be able to work with these a little bit later in the semester. So get excited